everyone, I'm Shura Sripal and we are discussing this question from GATE Examination 2023 paper Life Sciences question number 70. And the question states, the correct sequence of metabolic intermediates in Krebs cycle is A. Alpha ketoglutarate to fumarate to succinate to malate, B. Fumarate to malate to succinate to alpha ketoglutarate, C. Alpha ketoglutarate to succinate to fumarate to malate, and D. Succinate to alpha ketoglutarate to malate to fumarate. So for more information on Krebs cycle, you can follow this NPTEL course Biochemistry. The lecture is citric acid cycle and this is the YouTube link of the lecture. Now, Krebs cycle is a part of glucose metabolism. We know that Krebs cycle also has different other names like the tricarboxylic acid cycle or the TCA cycle, or it is also known as citric acid cycle. Krebs cycle is part of glucose metabolism and Krebs cycle only occurs inside the mitochondria. And we know that Krebs cycle is part of glucose metabolism when glucose is being metabolized aerobically. So it's a part of aerobic respiration. Now the glucose, so the first step in glucose metabolism is actually when glucose is converted to pyruvate by the process of glycolysis. Right, so glycolysis is the part or the steps or the reactions where glucose is sequentially converted to pyruvate, and this process occurs inside the cytoplasm. But this pyruvate actually enters into the mitochondria, and there it combines with the acetyl coenzyme A and it forms the first stable intermediate of the Krebs cycle, which is the citrate or citric acid. So which is why it is also called citric acid cycle, because the first stable intermediate of Krebs cycle is the citric acid or citrate. Now sequentially citrate will be converted and then finally it will come back to citrate again, which is why it actually operates cyclically, which is why it's called a Krebs cycle. And in this process there will be lots of energy equivalents that will be produced, like NADH, FADH2 and also ATP. So in this process, first of all, citrate will be converted to isocitrate, and there will be these reactions are catalyzed by specific enzymes. So citrate will be converted to isocitrate by the action of the enzyme aconitase. Now isocitrate will be converted to alpha ketoglutarate by catalyzed by the enzyme isocitrate dehydrogenase, and in this process one molecule of NADH will be produced, and citrate, which is a 4-carbon compound, will lose actually 1 carbon from it. So 1 carbon dioxide will also be eliminated in the process and it will form alpha ketoglutarate. Now alpha ketoglutarate will be converted to succinyl coenzyme A with the help of the enzyme alpha ketoglutarate dehydrogenase, and each of this time this dehydrogenation reaction is occurring, NAD plus or FAD will be converted to NADH or FADH2. So with these dehydrogenase enzymes, that is why they are important, because they are actually helping in producing the energy equivalents. Why are they called energy equivalents? Because NADH and FADH2, they will be finally coming in the they will be finally acting in the electron transport system, and there they will be acting as the carriers, the electron carriers. Now alpha ketoglutarate dehydrogenase catalyzes the formation of succinyl coenzyme A from alpha ketoglutarate. And here also you can see that one carbon dioxide molecule will be eliminated. Now succinyl coenzyme A will be converted to succinate with the help of this succinate dehydrogenase. Now succinate will be again converted to fumarate. In this process, FAD will be converted to FADH2. Now fumarate will be converted to malate with the catalyzed by this enzyme fumarase. So this is a water addition reaction where fumarate will be converted to malate. Now malate will be again dehydrogenased to form oxaloacetate. Now this oxaloacetate is actually formed from malate with the catalyzed by the enzyme malate dehydrogenase. Malate dehydrogenase also will actually add hydrogen to NAD plus and NADH will be formed, and this oxaloacetate will finally go on to form citrate, which will be formed with the addition of this acetyl coenzyme A and the oxaloacetate to form the citrate. Now this will be the whole process, and this will begin with the acetyl coenzyme A, right? And the first stable product of this cycle is citrate or citric acid. Okay, so now coming back to our question, what will be the correct sequence? So here it is given alpha ketoglutarate, fumarate, succinate and malate. So if we see that alpha ketoglutarate, succinate, fumarate and malate, this will be the correct order: alpha ketoglutarate, succinate, fumarate to malate, alpha ketoglutarate to succinate to fumarate to malate. So our correct option will be option C. Let's check it again once: alpha ketoglutarate to succinate to fumarate to malate. Alright, so that is our correct option, and that is why our correct answer is option C. That's it, and